Hello, welcome to Pharmacy 3030, the medicinal chemistry component of the course. So here's just a copyright message at the outset. So in today's materials, we're going to cover, there are two broad topics: insight into the drug discovery and development process, and then antibiotics, mechanisms of action, structure-activity relationships, and antibiotic resistance. Okay, so those are the two things. So the first is a very different series of materials to which you are used to me speaking about. So this is what we typically observe from a scientific perspective of barriers to drug delivery. We're trying to get our drug to a target here on the right hand side of the slide, and you start off with a dose of some description, say an oral dose, and then listed on this slide are all of the potential barriers that dose has to navigate to get all the way through to the site of action on the right hand side. And so there are many membranes, there are many pH effects that drug molecules have to survive past. They have to circumvent the liver and the enzymes responsible for phase 1 and phase 2 metabolism. They have to negotiate protein binding within blood. They have to negotiate the fact that they're going to be, whether or not they're going to be excreted into the kidney. Then they can be delivered through the barriers to the appropriate organ, distributed throughout the body, and then typically through passive diffusion absorb into the site of action, and they get from the cell to the particular target. And there are many barriers, and this is something that you're well aware of. But there are other barriers to drug delivery and drug discovery. And these relate to the more financial and bigger picture barriers of drug discovery. So this slide is all about getting to the top right hand side again. The idea is to try and generate a drug, and a drug that is approved for a particular indication. And so this slide has many metrics on it to explain how you can get there. So, for example, the biggest metrics in the box is how much it costs to typically take a drug from the discovery process at the outset to actually marketing a drug. Having it approved is about $1.75 billion. It's a large amount of money. Another way to consider the barrier to drug discovery process is the timeline. So along the bottom here we have from 0 to 16 years. It's split approximately into the first just over 8 years of drug discovery, and then subsequently the latter half will be the drug development process. You can also split the drug discovery process into the number of compounds that one has to screen to get a successful drug. So typically in the bottom right hand corner there's 100,000 compounds that are screened. That leads to about 250 molecules in preclinical studies, maybe 10 clinical trials, and if you're lucky, that will lead to one approved drug. Another way to view that is through the number of drug discovery programs that you have to start out with. So you have to start out with just over 25 drug discovery programs in the target to lead process, then developing a hit molecule into a lead, which means it's more drug-like. Maybe you'll succeed in about 20 of those programs. You have to optimize the structure of that lead so that it conforms to drug-like properties, so that it is suitable for preclinical trials, and maybe you'll end up with 15 programs at that point, and then you'll get preclinical success. The metabolism and the toxicity of the molecule are appropriate, and at that point, if you're successful, then you'll have about 12 programs that will get yourself into phase 1 clinical trials. And if your phase 1 is successful, and it's successful just over 50% of the time, you can then enter phase 2. Phase 2 demonstrates efficacy. That's about a third of the time they are successful. And then if that's successful, then 2/3 of the time you're successful in phase 3. And then you have the regulatory process where you have to submit the documentation to the agencies for approval, and ultimately you will end up with one drug. And usually this type of data relates to the US Food and Drug Administration, because that provides access to the largest market. And you can imagine, if you're going to spend $1.75 billion on making a drug, you want to recoup that as quickly as possible. And you do that by selling that drug in the largest market. And that, to this day, remains the US healthcare market. So I've put in here 3 dates relating to drug regulation and development, which situate the drug discovery and the drug development process nowadays. So the first is back in 1906, the Pure Food and Drug Act was enacted in the US, and that ensured that people had to label everything appropriately. So if you were selling something, you had to say what it was you were selling, and it had to be what you were saying. And if that wasn't the case, you could be prosecuted. Then in the mid-1930s, the Food, Drug and Cosmetic Act came into play, and that was an act that meant that medications had to be safe. And that was brought about by a disaster where sulfanilamide, which was a synthetic antibacterial, was being sold, but it was sold as a solution in diethylene glycol. And that resulted in over 100 people dying because of the diethylene glycol, because that's toxic to our bodies. And so that tragedy meant that the Food, Drug and Cosmetic Act was brought in. And then the final date that I want to, or act that I want to remind everybody of is the the Drug Efficacy Amendment. And this meant that drugs had to undergo clinical trials to demonstrate efficacy in humans. And this was brought about because of the responses to thalidomide. So thalidomide was a drug that was taken by women who were expecting in an attempt to stave off morning sickness. The tragedy is that many of those mothers gave birth to children who had birth defects, and those defects were ultimately demonstrated to be due to the taking of thalidomide. And it was due to one particular scientist at the FDA who reviewed the data on thalidomide in the European Union, or it wasn't in Europe, I guess it wasn't the European Union at the time, and she said that it wouldn't be appropriate to approve that drug in the US, and she had to stand up against considerable pressure to say no, it's not safe to have that drug in the US. And obviously she was vindicated by her stance based on the data that she was able to demonstrate to people. And so that, as a result of that thalidomide those thalidomide tragedies in Europe, and the Drug Efficacy Amendment was brought in, meant that human drugs had to be subject to human clinical trials. And so most of the data that I'm presenting comes from this particular textbook. Most of the slides come from that textbook. So this is another overview of the drug discovery and development process. It's the timeline is a little bit different on the x-axis here, but the process remains the same. You have a discovery stage where you take a hit molecule into a clinical candidate, and then development stage where that clinical candidate is converted into clinical trials and ultimately a drug. So drug discovery is expensive, and how do you recoup the costs? So this is the rationalization for patenting in drug discovery and development. So intellectual property is the solution. And so there are 4 types of intellectual property, or broadly speaking. There is copyright, that's what I have at the start of my slide, saying that this is materials that I've generated and put together. You can have intellectual property in the term of trademarks. There is a particular, Dalhousie has a particular crest and logo that is Dalhousie's trademark. There are trade secrets. Maybe that's the formula for Coca-Cola. It's never written down, nobody knows what it is, but certain people know certain aspects of it, and that makes the unique Coca-Cola formula. But the form of intellectual property that is most valuable, I mean, trade secrets are very valuable, but you have to keep them secret, obviously. The most important commercial intellectual property are patents. And so patents are an agreement between the recipient of the patent and the government, or the inventor and the government. And it is a document that provides you as the inventor the right to exclude others from making, using, offering for sale, or selling the invention or importing the invention. And the bottom line, the caveat is for a limited time, and you have to exchange how you do it That's the public disclosure of the invention. Okay, so a patent document describes how it is you, in this instance, would make a drug and what that drug is good for. In return for disclosing that, you have the right to prosecute anybody else from making or selling that particular drug in the country where the patent is issued. Okay, and so as a consequence of that, that right allows the patent holder or somebody to whom you license the patent to the opportunity to recoup the investment made in the lengthy and expensive drug discovery and development process, and then to make a profit. And sometimes they can be big profits, and sometimes you don't make a profit at all. In some cases, the patent is held by an organization such as a university and is then licensed to a commercial entity such as a company to complete the drug development process. So generic drugs that are sold by commercial organizations other than the original patenter or licensee, that they are enabled after patents have expired, and they're termed generics. After the patent has expired, the drug can be sold by anybody. You have to demonstrate bioequivalence of the product, and the regulatory agencies have to approve that application. But then once that's been done, you can sell it. So therefore, generic firms do not need to support the drug discovery research efforts, and therefore their costs of running a business are much less. And so they can sell a generic drug for much less. So what is a patent and how long does it take to get one? So it takes a couple of years to get a patent. You file patents concurrent with the drug discovery and early preclinical development stages. Patents are comprised of claims of novelty, and you submit the document to a patent office, and that office is a government agency, and the patent is then examined by a patent officer of the government. The examiner reviews the literature and other patents to confirm the novelty, and they can request changes or reject certain claims. In the case where multiple patents are filed on the same subject matter, it is the first to file as the inventor, as opposed to the person who first invented something, and that's a relatively recent change to law. Once a patent is conferred, the government office is not responsible for enforcing the patent. That responsibility falls to the patent holder. Typically, multiple versions of patents are filed. So for example, it could be the composition of matter. That means the unique structure of the chemical or the biological entity. The method of using a material. So minoxidil, it was a heart-lowering drug initially, and then it was discovered that it promoted hair growth. So the hair growth discovery was a second patent. How to make something, a process patent. You can prevent others from using the same method to make it, and you can patent that approach. How you formulate something, that is obviously can be unique, and therefore that's patentable. So patent lifetimes typically last 20 years, and patent occurs towards the start of the drug discovery process. So if it takes anywhere between 8 and 12 years to get a drug to be approved, then you only have between 8 and 12 years to make a profit. Or rather, you have exclusive right to sell the drug, which is when you're going to recoup most of your costs. So preclinical studies include process chemistry research. So that's how you're going to make something to be safe and efficient. And the point here is that we're making large amounts of material, and the number of operations need to be minimized. It needs to be an environmentally safe process. It needs to be efficient, highly reproducible. The yields and the impurity profiles have to be high yields and minimal impurities. And so just to note that how you make something initially for the drug discovery process and then how it is made when it's a much more valuable commodity are very different. Okay, when you're making hundreds of thousands of compounds, the most important thing is to make it. But when you're only making one or two, then you need to make it very efficiently. Okay, the types of chemical reagents that you can use will vary depending on the scale of the reaction, how you're going to purify the material, the stability of the materials you're going to use, and how you go about all of this can all be patented, and that can extend your patent lifetime and exclusivity for a particular drug. You can also, during the preclinical trial process, have drug toxicity determination. So things are poisons. You need to get the right dose, you need to determine the therapeutic index for a particular drug. Obviously, you want that to be the highest tolerated dose and the minimally efficacious dose, and that ratio should be as high as possible. But it does depend on the type of disease. So for non-life-threatening diseases, you need a really high therapeutic index. But for a life-threatening disorder, that index can come down substantially. Because I guess the point is, if there is no treatment and it's a terminal disease, then regulatory agencies are more inclined to say, yes, you can try that particular medication. Toxicity. Preclinical studies involve various assays to confirm that the molecule is not toxic or cytotoxic to human cells, to make sure that molecules are not going to be involved or induce cytochrome P450 inhibition, that molecules are not going to bind or interrupt the hERG channel receptors in the heart and generate arrhythmias. And then obviously additional preclinical studies include genetic toxicity to assure there is no mutagenic potential of a particular drug. Those preclinical studies, the toxicity testing is done with in vivo assays and then subsequently in multiple animal species. So you do lead optimization and pharmacokinetic studies in vivo, or after in vitro studies. You do acute toxicity studies in animals at increasing doses until you see what that toxicity level is. And then you do range finding studies to confer different doses over longer periods of time. And then when you're getting closer to a clinical trial and you're working out how long a clinical trial is going to be, you do subchronic testing or chronic testing of the drug. So dose the drug repeatedly up to the length of that clinical trial and ensure that in animal studies that there are no observable toxicity issues. And then also see what the issues are in terms of reproductive fertility, because clearly you'll need to be able to show whether or not there will be effect on the progeny of parents. Preclinical studies also include formulation of the final drug product. So the active pharmaceutical ingredient, that's the bit that as a chemist I'm always so excited about. But the reality is there are many other components in a pill, a capsule, or a liquid. Those excipients are molecules in their own right that have significant effects on the degree of absorption, the half-life, the bioavailability, and the dosing schedule, how easily a drug is released. They can also affect the stability of the molecule to promote its inertness to heat or to humidity or to pH, and constantly deliver the same drug levels over a period of time. Formulation factors can often be related to the physical properties of the drug molecule, such as its solid state form, the crystalline polymorph, the particle size, the pKa, the solubility, the type of salt form, how rapidly it dissolves, and all of these factors that affect a molecule and its formulation are all patentable. Okay, if a drug has low permeability, formulation doesn't really solve the issue. That sort of thing. So all of those are the types of things that you can patent, and with all of those patents, they generate you exclusivity for your novel drug. So once you have filed all of the preclinical, done all of the preclinical research, filed the appropriate patents, then you identify a clinical drug candidate, and it would be the molecule that exhibits the best potency, physical and pharmaceutical properties, and low toxicity. And you submit all of that data to the US Food and Drug Administration as a new drug application. And that IND, or investigational new drug application, and that IND needs to be approved by FDA prior to clinical trials. Obviously, you would then also require ethics approval for the clinical trial, but the ethics approval would be by the university body that is going to facilitate those clinical trials. And then you embark into clinical trials. So we start off with phase one clinical trials, typically a small number of healthy volunteers, and you're looking for unexpected toxic effects. So you're looking whether or not that molecule is safe. There is no requirement for efficacy at this stage. If the molecule is safe, and that's always the first thing that regulators want, the molecule needs to be safe. Once you've demonstrated safety, then you can compare efficacy to either a placebo or the best-in-class medication that's out there. Often that type of study would then be double-blinded. And then if you've demonstrated efficacy in a phase 2 study, you then move into phase 3, which is a larger study, wider patient population, that potentially have combinations with other drugs. You're looking at the tolerability of a particular medication and, as ever, the safety of that drug. And then all of this data and the preclinical data are submitted as a new drug application to the FDA or to Health Canada. And then if that molecule is approved, then the drug can be marketed based on what the regulatory agencies say. Okay, so you can only market the drug for the disease for which you have demonstrated efficacy. And there can also be a requirement for post-marketing surveillance, where the drug agency can say, given the data you've presented, we would like you to continuously monitor patients for this particular issue, because there may be some problem in the future, and we need you to present the results of that study to us in a couple of years' time to confirm or to refute whether or not the drug is sufficiently safe. And so that can be a requirement before the company would be allowed to market and sell the drug. So that's the end of the materials for the patenting and the clinical trials, the drug discovery development perspective. Next, we're going to move on to antibiotics. So the antibiotics part of this is really a summary of everything that you did previously in the second year and a little bit more besides. So we start off at the top with the structure of a bacterium, and just highlighting some of the structural features where that drugs target, and the major structural features of a bacteria. And then below that we have the gram-positive cell wall and a gram-negative cell wall, and the differences between the two, which is the justification as to why drugs target either gram-positive or gram-negative organisms. Typically, it's easier to target drugs against gram-positive organisms than gram-negative, and that's because of the additional outer membrane on a gram-negative organism, and also the O polysaccharide, or the lipid A, that is present in a gram-negative organism. Then there are a couple of terms that we have to look at: virulence and resistance. So bacteria multiply outside of cells, and they can be classified as pathogenic, opportunistic pathogens, or non-pathogens. And so the virulence is a measure of their pathogenicity. So how well our own immune system is able to deal with a particular organism. Okay, so a pathogenic organism is one that is always going to cause an infection. An opportunistic pathogen is one that our immune system is usually able to deal with, but if we are immune compromised in a particular situation, then the organism can then successfully infect a human. And then there are non-pathogens, bacteria, that do not cause disease. Resistance to antibiotics come about by a number of mechanisms: production of drug inactivating enzymes, evolution of the altered drug targets with reduced drug affinity, decreased permeability of the bacterium to drugs, and production of drug efflux systems. Okay, here is a cartoon representation of some antibiotic resistance strategies that exist. And so this is the graphical representation of what I've just said. So there are inactivating enzymes that will cleave our drug molecule or append some different functionality to our drug molecule. We have efflux pumps that, if the drugs got into the cell, then they can pump it out, so the concentration is never high enough. Maybe an alternative enzyme pathway is switched on to convert substrates to products. So even if our drug shuts down one pathway, another pathway exists. There are, depending on how a drug gets into a cell, you can have decreased uptake, and then you can also alter the target. So drugs that target the ribosome can be modified so the drug no longer binds, or if a drug targets DNA or an enzyme that processes DNA, that enzyme can be mutated such that the drug no longer binds. So antibacterial drug discovery started many years ago long before we were around. And Ehrlich was the scientist who was at the time looking for a treatment of syphilis, and he discovered salvarsan. And I just put this in here not because, because I guess you're doing the STD unit, but also because this is the structure of the molecule that was proposed in 1910, and in 2005, so just 95 years later, the structure was revised to these two molecules here. And so I just put that in to say that you don't have to have the correct structure to have an approved drug. Providing it's safe and providing it does what you want it to do, then you can get away with having an incorrect structure. I mean, clearly they did, to the best of their knowledge, they confirmed the structure here, and you can look and see that it has all of the same structural components. It's just the various ratios in the bonding type that is different. Anyhow, antibacterial drug discovery then went through discovering molecules such as proflavine, which would intercalate into DNA, into sulfanilamide, which is an inhibitor of part of the folic acid biosynthetic pathway, and then penicillin G is an inhibitor of the penicillin binding proteins in the 1940s. And so those discoveries really spearheaded the antibacterial discovery process. Nowadays, there are a selection of pathogens for which better antibiotics are needed because these pathogens have developed resistance to most drugs out there. And so there are, they have been labeled the escape pathogens based on the abbreviations of the first letter, which is a great way of remembering what's going on. So there's Enterococcus, there's Staphylococcus, there's Klebsiella, Acinetobacter, and Pseudomonas and Enterobacter species. So these all exist, and they all have drug-resistant forms, and in hospitals they can cause substantial problems. Bearing in mind that in Nova Scotia the antibacteriogram data exists, so you will know more precisely the types of organisms for which resistance has developed within Nova Scotia, and clearly that data is more important than what I say is a generic understanding of the drug resistance process, particularly obviously when you're in clinical practice. So we're going to run through categories of antibacterial drugs, and we're going to base them on the mechanism of action. So we're going to have cell wall inhibitors, protein synthesis inhibitors, nucleic acid synthesis inhibitors, and then lastly drugs that affect membrane permeability. And obviously we want something that, ideally we would like something that targets gram-positive and gram-negative bacteria that is orally active with low toxicity, but we don't get all of that in one pill very often. Okay, we'll start off with cell wall synthesis inhibitors. So the penicillin binding protein or the transpeptidase is responsible for catalyzing this particular reaction. And so we have NAG-NAM units that are part of the peptidoglycan, and the cross-linking process takes the molecule on the left-hand side here, the penicillin binding protein that I've just highlighted, covalently binds to the cell wall through this ester linkage with the release of the dealanine residue, and then a amino functionality from the other component of the cell wall comes together to form a new amide bond. And that's the linkage that the transpeptidase catalyzes the formation of, and so we have then cross-linked the peptidoglycan. So to summarize that reaction, it's on the top half of this slide. So the structure here is the dealanine, which is just the terminal fragment of the peptidoglycan. And we have a particular serine hydroxyl side chain that acts as a nucleophile on the penicillin binding protein. It attacks the amide. We form this tetrahedral intermediate. That sp3 center breaks down to the sp2 ester-linked penicillin binding protein. And so then this species here then reacts with another component of the cell wall, comes in and attacks here. And the scheme stops at this point because below that we show how penicillin G reacts with the penicillin binding protein. So shown here is the structure of penicillin G. You see the beta-lactam ring, which is the cyclic amide functionality. How when that cyclic amide reacts with the transpeptidase, we form a sp3 center, the tetrahedral intermediate. It collapses, so we reform the carbonyl group and cleave the carbon-nitrogen bond to generate an ester linkage here. But because of the fact that we have the ring system, the beta-lactam, this component part on the right-hand side does not depart the enzyme active site, and it blocks a water molecule or the other component of the cell wall coming in to react with the acylated enzyme. And so that stops the penicillin binding protein catalyzing any more chemistry. So that's the way that the penicillin enzymes block the function of the penicillin binding proteins. In terms of structure activity, the beta-lactam ring is required for all of these molecules. We have the 5-membered ring with the sulfur and then the dimethyl substituent and the carboxylic acid. The carboxylic acid mimics the carboxylate of the dealanine functionality. So early penicillins include penicillin V, G, and methicillin, which is a good molecule to use to block beta-lactamase resistant bacteria. They're typically made by obtaining 6-amino penicillinic acid, and then you can modify that amino group with many different functionalities. There are many varieties now of penicillins, and so shown here are just some examples. So we have more beta-lactamase resistant molecules at the top, and they block the reaction of the beta-lactamase with the beta-lactam ring. The amoxicillin and ampicillin have broader spectrum of activities, and that's suggested by the presence of this amino functionality. Then we have other derivatives, carboxypenicillins down below that, and then the uridopenicillins have more modified functionality based on ampicillin, where we have the amino functionality has been modified into a urea derivative with modified functionality that gives rise to drugs that have more specific diseases that they treat. And then we have to look at what beta-lactamases do. And so beta-lactamases are the enzymes that cleave the beta-lactam ring. And so here we have our beta-lactamase, here we have the enzyme nucleophile, so they have a serine in the active site, just the same as the transpeptidase. And the first thing that happens is you generate that tetrahedral intermediate, you get a stabilization of that negative charge, and then we cleave the carbon-nitrogen bond to give an ester linkage, and so we have our acyl enzyme intermediate. And so that's very similar chemistry to what we see with the penicillin binding proteins. The difference, however, is that with the beta-lactamase, this ester functionality remains accessible to hydrolysis. And so our water molecule is able to enter the active site despite the presence of the other component of the 5-membered ring and the carboxylic acid. That water molecule attacks the ester functionality, forms another tetrahedral intermediate, which collapses down to release the active enzyme and the hydrolyzed beta-lactam. And therefore, that molecule of beta-lactam is no longer going to be active. That penicillin or cephalosporin is no longer active. Thankfully, Mother Nature came up with an example of a beta-lactamase inhibitor. So that's clavulanic acid. Other synthetic derivatives, I think, such as tazobactam and sulbactam exist. And so these molecules react with the beta-lactamase, and they forming an ester linkage as shown here. But then because of the nature of the different 5-membered ring, we get a rearrangement process occurs, releasing the carbonyl group here and the imine functionality. And it just so turns out that that imine functionality will react with another hydroxyl on the enzyme to generate this bridged inactivated beta-lactamase. Okay, and so then with the two covalent bonds holding the fragment of the drug, the beta-lactamase has been shut down and is no longer able to hydrolyze the penicillins or cephalosporins. So cephalosporins are beta-lactams that target more gram-negative organisms. They differ from the penicillins because they have a 6-membered ring versus a 5-membered ring on the right-hand side. And typically there are 2 places of structural modification permissible for the cephalosporins. We have the same R functionality on the left-hand side, but also this R' functionality on the right. Increasing generations of cephalosporins typically improve gram-negative activity at a cost of lesser gram-positive activity over time. And resistance to penicillin cephalosporins occurs through production of beta-lactamases and also through the production of mutant porins, which are the proteins responsible for enabling molecules to pass through the cell membrane. And when they're mutated, they no longer emit cephalosporins, and so there is insufficient concentration of the cephalosporin inside the cell to cause activity. So here are some examples of the generations of the cephalosporins. And so it goes that you can recognize some of these. There are some functionality that are consistent with beta-lactams, with penicillins. Cephalexin has a similar side chain here to ampicillin, but then there are some different derivatives. Cephalothin and cephozolin have these different functionalities. You can see a couple of those present in some 2nd-generation cephalosporins. The substituent highlighted in red here helps block beta-lactamases hydrolyzing the amide bond. 3rd-generation cephalosporins, the functionality on the left-hand side has got larger and more elaborate, and typically has this 5-membered ring system present, shown there. And then the functionality on the right-hand side also varies as well. And then down at the bottom we have some 4th and 5th-generation cephalosporins. So we have that similar functionality on the left, and it is typically the part on the right that is varied. So we have some positively charged nitrogen functionality for all 3 of the drugs, and this one also has a 2nd ring extending where that positive charge is versus the beta-lactam core. Those aren't the only beta-lactam types of drugs that exist. Thionomycin was a natural product that was isolated many years ago. It's too unstable, but there are a variety of derivatives that have been prepared that are used clinically. Imipenem is one of those, and so you can see how it's modified relative to the natural product. Note that it's similar to beta-lactam in that we have a 4-membered ring fused to a 5-membered ring, except with that 5-membered ring we have a carbon atom at the top, versus previously we'd have had an oxygen or a sulfur for the previous derivatives we've discussed. Monobactams also exist, and these are more potent towards gram-negative bacteria, and so these just contain that 4-membered ring, and we would anticipate that these would react with penicillin binding proteins in the same manner to described previously. So then switching to glycopeptide antibiotics. So these are molecules such as vancomycin, dalbavancin, and teicoplanin. So these are complicated molecules. The differences in terms of the structure is highlighted in red. And so we have a peptide backbone through the middle, and off that peptide backbone we have different functionality, so non-proteinogenic amino acids that are typically linked together through ether linkages or through carbon-carbon bonds. And that gives rise to these molecules being sort of glove-like in terms of their shape, like a bit like a baseball glove. And it is the pocket inside that glove that they bind a dealanine, which is part of the cell wall peptidoglycan. And so these molecules target primarily gram-positive organisms. And so shown on the left, we have in blue the structure of vancomycin, and shown in green is the dealanine peptide, and in dotted lines are the hydrogen bonding interactions between that peptide chain and the vancomycin functionality. And those hydrogen bonding interactions have been drawn out on the structure on the right, indicating which atoms are responsible for those hydrogen bonding interactions. In the scheme on the right, highlighted in red, is a particular NH, and that's highlighted because in some vancomycin-resistant organisms, those that express the VAN A to X and RS genes, the NH is switched to an oxygen atom. Okay, so and therefore that means instead of having D-ala as the two final components of the cell wall, so there is one alanine residue and there is the other alanine residue, there we go. So it just has the one stereocenter with the methyl group around it. That first D-alanine residue is converted to a D-lac, which means that we have an oxygen replacing the NH. And as a consequence of putting in that oxygen, we are unable to have a hydrogen bond between that NH and the carbonyl group here, because we have obviously an oxygen replacing the NH. And that lack of that hydrogen bond means that there is about 1,000-fold reduction in activity or affinity for vancomycin for the modified cell wall. And so those vancomycin drugs are incapable of shutting down the bacteria. Next, we're going to look at bacterial protein synthesis inhibitors. So this is a cartoon representation of what the ribosome is. It's the large purple-like blob responsible for binding an mRNA molecule at the bottom, which encodes for a particular protein. The ribosome then binds the appropriate tRNA, and then the peptide is transferred from the P site to the A site. So the K, the lysine here, is then bound to the serine to extend the peptide, and then the ribosome moves along, and then the tRNA binding here, the peptide, so the serine residue will then form a new amide bond with the valine residue, and so the peptide gets longer. So there are many drugs that target the ribosome. The ribosome is a very large, complicated structure of proteins and RNA molecules. Molecules that target it include aminoglycosides, tetracyclines, and macrolides. So they're all natural products, and there are some synthetic or semi-synthetic derivatives thereof. Aminoglycosides are shown on this particular slide, and so shown in red is the functionality that is the six-membered amino hydroxylated ring, and that's the functionality that is consistent between all of the aminoglycosides. As you can see, they then have various amino sugars appended to them in some capacity, and that enables them to bind the ribosome specifically. They're very polar molecules, all of the aminoglycosides. And so shown here is a cartoon an interaction diagram on the left showing how streptomycin binds to the 30S bacterial ribosomal subunit. And so there are lots of polar interactions between the drug and the various phosphates present in the active site of the ribosome. And then on the panel B is just the stereo diagram from the literature where the data was published, showing how the drug fits into a cavity where there are all of these different helices from the 30S ribosomal subunit that interact around the drug to give the active site pocket where the aminoglycoside binds. And so it does so, and it stabilizes what's called a narrow-prone form of the tRNA in the A site, and it therefore the binding of the streptomycin and the aminoglycosides leads to mistakes in the translation of the proteins and shutdown of protein synthesis. Tetracyclines are another class of antibiotics, have the structures down below, and typically they have a binding interface to the ribosome, which are the hydroxyl functionalities, and their structure can be modified on the opposite face of the molecule. So the R functionality here can be modified for the different tetracycline derivatives. And tigacycline is an example of a glycol cycline, which is a much more recent semi-synthetic derivative that's been discovered, where this additional functionality shown in red extends how they bind to the ribosome, and also they bind in such a way that they stop some previously drug-resistant forms of tetracycline. So they're able to bind and shut down bacteria that were otherwise resistant to tetracycline. So this is how the drugs bind to the ribosome. So it's that polyhydroxylated face. They have a coordination through a magnesium ion to the polyphosphate backbone. And so that's how the drug interacts with the ribosome, noting that the side where the structural modifications occur points out into space. And then down below that is just the stereo diagram from the literature that shows that where the magnesium ion is, that's the species that I've just highlighted, where the hydroxyl functionalities coordinate to the ribosome, and then we have all four of those rings, and then the back four of those rings point into space. So there is the possibility for structural modification in those particular sites because there are no interactions between the drug and the binding site. Next we have macrolide antibiotics. They also bind the ribosome, and I think I have a slide moving on that shows how they will bind the ribosome. But first, just some examples of the structures of the macrolide antibiotics. So these have a large macrocycle about them with a ester linkage or a lactone functionality. This slide just shows that in an acid environment, erythromycin reacts to form, first of all, this hemiacetal, which then cyclizes to form an acetal, and then at this point the drug is inactive and it changes the shape of the molecule sufficiently that it no longer binds the ribosome. Derivatives that have been made to circumvent that are shown here, and it's just that they're very rational changes. So the derivative clarithromycin, if we have this methoxy substituent here, remember this hydroxyl is involved in attacking the carbonyl group just here. So when we've made the ether functionality, that can no longer happen. So the acid hydrolysis does not occur. We have azithromycin, where the carbonyl group has been converted to this tertiary amine. That means that the acid hydrolysis no longer occurs. The presence of that amine also gives rise to very different pharmacokinetic properties for azithromycin that often give rise to much longer dosing regimens for azithromycin, so you don't need to take it multiple times a day, potentially even only just once a week. Oxithromycin is another derivative where the carbonyl group has been modified such that it is going to be acid stable, and in addition we have the methoxy substituent there. Another example is telithromycin. This derivative, we have the methoxy substituent here, so that stops the acid insensitivity, and we also have this carbonate functionality installed with this aliphatic chain and the aromatic functionality on the end of it that extend out into space. So here is an interaction diagram showing how these drugs bind the ribosome. Suffice it to say, it's pretty complicated. I'm not expecting you to remember the numbers, but what you will note is that there are many polar interactions. So hydrogen bonding interactions are key between the drug and the ribosome, and that's not surprising when the ribosome is such a polar entity, and the drugs themselves have so many hydroxyl functionalities present on them that can form hydrogen bonding interactions. Another group of drugs are the pleuromutilins. These also bind the ribosome. They bind the 23S site, and they inhibit the peptidyl transferase reactions. That's the transfer of the polypeptide chain. So making that amide bond, they stop that process. So shown on the right-hand side is how the drugs fit into the active site or into the ribosome. And so the reason why I've showed this particular structure is that all of the functionality that remains the same, if you like, the business end, it sticks down into the cleft where the ribosome loops around, and then extending out from that is the functionality that can be varied. And so all of those structural analogues are shown here, so you can see where the structure changes. So here we have the hydroxyl or this side chain, or the ring species here, or lefamutylin is this derivative with this six-membered ring with the amino hydroxyl, and that's the one that's shown bound here, and so I've just circled that functionality also. And so I put in this example of the pleuromutilins because lefamutylin was approved for human use within the past couple of years. But what's interesting is that after all of the drug discovery, and the need for antibiotics that are complementary and able to treat drug-resistant bacteria, the sales of lefamotylin were very poor last year. So it barely made several $100,000. Yet you know how much it costs to bring a drug to market. And so it's just an interesting perspective of somebody made the effort of bringing a new antibiotic to market, but at the moment they're not going to be able to recoup their costs of that drug discovery process because it's not being used sufficiently. Finally, the last group of drugs that target the ribosome are oxazolidinones. And so the example here is linezolid, and it fits into a pocket of the ribosome. So it blocks the tRNA from entering the ribosome. And so shown here is the ribosome in grey, and it forms a sort of shape around here. There is a very unique kink to linezolid between around the stereocenter through here that is represented here, and it's that kink that mimics the shape of the ribosome, how the drug fits in very nicely. And I should say that linezolid was approved, or a new class of antibiotics discovered in about 2000 and approved. And at the time it was quite, people were quite excited because it was the first new class of antibiotics for maybe 30 years. But unfortunately, drug resistance developed relatively quickly thereafter. Next, we're going to move on to some drugs that target antibacterial agents that inhibit the DNA gyrase. So these would be the quinolone type drugs. They do not bind directly to DNA, but they bind a DNA gyrase complex. And so the structure activity is shown in the slide here. And so they're based on nalidixic acid. So these compounds are synthetic molecules, so where you can modify the functionality around the diaryl framework. The carboxylic acid and the carbonyl group are always present. The R functionality at position 6 is almost always a fluorine in the more active drugs, and then R7 is where there is a greater diversity in structural modification. And so some of those examples are shown on the next slide. Some second generation quinolones or fluoroquinolones, as they're showed. Ciprofloxacin is the classic, and then fourth generation derivatives down below, so you can see how they are similar yet also differ. There are some structural features that remain consistent between them and some of the second generation, and then some of the functionality that differs between them also. And so those subtle changes, they don't change the target, they just change the properties of the drug. And so this just shows how the quinolones bind to the topoisomerase. So the representation on the top here in yellow, we have DNA double helix. We have the drug shown as a space filling the blobs in here, and it's the topoisomerase that is in green and blue. And the topoisomerase binds covalently to the DNA as the DNA is unwound, and the drug fits within that. And the picture in the bottom left-hand side shows how a magnesium ion is responsible to coordinate between the carboxylic acid, the carbonyl group, and some water molecules and specific amino acid residues present on the enzyme itself. And so you can see that, again, that's how the drug coordinates to the target. And so it makes sense that the structural variation occurs in the opposite side of the molecule. Okay, and so that can improve the properties of the drug so that it can better penetrate a different organism, for example. Next, we're going to look at folic acid biosynthesis and the sulfonamides. So these were some of the first synthetic antibiotics ever discovered. The drug discovery story is always a nice one because this particular dye, prontosil, is a bright red color, I presume. Azoreductases in the gut reduce the N double bond N to an amino functionality, and this is the compound that is the antibiotic, and it mimics the para-amino benzoic acid in the folic acid biosynthesis. And so shown below as analog sulfur methoxazole that is used clinically nowadays, particularly in combination with trimethoprim. So the sulfonamides and trimethoprim inhibit the folic acid biosynthesis. The para-aminobenzoic acid reacts with dihydropterate diphosphate with an enzyme called dihydropterate synthase to form this new carbon-nitrogen bond just here, and then subsequently the aspartic acid is installed to form tetrahydrofolic acid, and so the sulfonamide functionality replaces para-aminobenzoic acid, and that's shown in the slide down below. So it forms that same carbon-nitrogen bond, but then when you have the sulfonamide functionality present here, the coupling to the amino functionality of the amino acid is no longer property, so you no longer are able to produce folic acid. And remember, folic acid is essential for bacteria to survive, so if they cannot produce folic acid, then those bacteria are going to die. And remember, as humans, we have to take up folic acid as part of our diet, so compounds that inhibit the folic acid pathway are not going to be toxic to ourselves in terms of, because we have no folic acid pathway in our bodies. Trimethoprim inhibits the production of dihydropterate diphosphate, so it binds to the dihydropterate synthase enzyme. So it binds, and you can imagine that the aromatic functionality mimics the ring systems. Finally, we're going to move just a couple of slides on drugs that affect the bacterial cell membrane permeability. So typically these would be cationic peptides that have a somewhat non-specific mechanism of action. Okay, they are toxic natural products. They've been known since the 1940s. They're good veterinary medicines, but they've been sufficiently developed now in such ways that they can be used for humans. So these are cyclic peptides. They displace calcium and magnesium ions from membranes, destabilizing the membranes. They contain fatty acid tails that interact with the LPS and allow membrane leakage of the cellular components. So here are some examples. So colistin or polymyxin B are the examples. Daptomycin is the most recently approved analog. So we have this cyclic peptide that is connected through more amino acids to a fatty acid tail, and it is the assembly of this to bind the metal ions that then associates with the cell membrane and disrupts the cell membrane. Finally, I just want to say the types of exam questions, given that this is an online course. So the MedChem exam is going to be open book, so you can look at your notes. You're obviously not allowed to communicate about the exam with any person or some website. The chemistry questions, I've given a couple of examples after this so that you know how they're going to be formed. So they're all going to be 1 point multiple choice. There will be an image that will be a part of a question, and then with a series of potential answers. So shown here is one example. So this drug is most likely A, and then you have to identify the type of molecule that it would be. So which A, B, C, or D you would anticipate this particular molecule would be. And then another example of a question. So here we have a very similar molecule. Which functionality is essential for the mechanism of action? And so you would then have to choose whether it would be functionality 3, functionality 2, functionality 1, or functionality 4. And then you would be able to select your answer appropriately and move on to the next question. Thanks very much.